All right, so we've recognized that there's a problem. We've shouted for help. Those are the first two steps that innate immune cells will take in a response to a pathogen. They have to know kind of what the problem is using their PRRs, their senses, to kind of figure out what type of help they need and shout for it. So now what's going to happen next? Well, the next thing is everything will hopefully come together where it needs to be. Now, the process of convergence is actually known as chemoattraction. And this is basically kind of a catch-all term for all of the things that occur to get the white blood cells, or almost any cell, but for here, we're going to be focusing on leukocytes, the proper term for a white blood cell, and getting that leukocyte where it needs to be, which is the site of infection. So we had that cytokine release, we've had that shout, and I warned you that cytokines would come up again. Not only do cytokines alert the other cells that are in the area, the non-immune cells, cytokines are going to play an important role in getting everything ready so that the cells can get where they need to be, the white blood cells. And one of the first things cytokines is going to do is that when it is talking to the non-immune cells, when it is talking to the endothelial cells that line the blood vessel, it's actually going to tell them to let the blood kind of leak out. This sounds a little weird. Why would we want increased vascular permeability? Increased vascular permeability is actually what leads to inflammation. The blood leaking out of our vessels and into the tissue, in particular into the skin, but into any tissue, is what leads to the heat, redness, swelling, and pain of inflammation, the calor, tubor, tumor, and dolor of inflammation. Heat, just because there's a lot of the blood, which is coming from our core out to our extremities. The redness, because some of the red blood cells are going to be leaking out into the tissue as well. The swelling, because we have fluid there. And pain, why? Well, we have a lot going on. We're going to have a lot of indication of a problem. And this pain is actually a good thing. I know we always think of pain as bad, but it warns us. If we cut ourselves and didn't even realize, we wouldn't know to sort of wash it out, to tape it up, to avoid using it. So pain is actually beneficial here. But let's talk a little bit more about that swelling and that fluid coming in. That's where sort of the good stuff is happening, good when it comes to immune responses, at least. The leakiness of that blood vessel means that it is going to be easier for cells to get through that blood vessel and into the tissue. And we'll talk about how the leukocytes do that in a second. But almost as important is just the fact that, remember, we have all these non-cellular components of innate immunity. We talked a lot about complement, and there are some other proteins like the C-reactive protein that are just floating through our blood, produced by our liver there. And that complement and those C-reactive proteins, they themselves, as they're leaking out of the blood into the tissue, can help start to fight off the pathogens, in particular bacteria, viruses, fungi, possibly even helminths and other parasites, even before we can get the cells there. So increased vascular permeability triggered by the cytokine release. Still focusing on cytokines, so we're actually going to go to a special subset of cytokines. So if we considered cytokines as a huge group of communication molecules, chemokines are a more specific group of communication molecules that have to do with chemoattraction. Kind of using a lot of these terms here. Chemoattraction, all of the steps that are needed to get the leukocytes to the site of infection. The chemokines are the cytokines that are signaling to those cells. And you can see here in this figure, cytokines have all different types of names: INF, interferon, TNF, tumor necrosis factor. And there are some habits. You'll notice that immunologists are awful about abbreviations. We can't just say interferon, we say INF. But that's also helpful because then we talk about the INF receptor. So INF is a signal, the receptor is what hears it. TNF and TNFR, tumor necrosis factor and tumor necrosis factor receptor. Chemokines are going to have their own special abbreviations. They're generally going to be CCL or CXCL. And that has to do with their protein motifs. We're not really going to get into it. But CCLs and CXCLs are the chemokines themselves. The Ls are for ligand. The R's then on the cells, the CCRs and CXCRs, those are going to be the receptors. So as we're starting to get into things, you may see a lot of CCLs and a lot of their receptors, the CCLRs or CCRs, not the L's. All of these are signals and receptors for basically directions. They're communicating directions of where you need to be. And the first thing that these chemokines are going to do is go into the bloodstream. Remember, we already have the leaky vasculature. So those cells that were releasing and screaming, that signal is going to go into the blood. And it's going to reach the immune cells that are hanging out in the reserve spaces. We don't want a ton of leukocytes and immune cells. Leukocytes are immune cells in our blood. That would make our blood really thick. It would make our heart working so hard. So we basically keep them on standby, largely in the bone marrow, to some extent in the spleen and a few other places. But when chemokines are flowing through the blood, these leukocytes are going to get a signal that it's time to get out of the bone marrow, come into the blood, because they are needed in the tissue. So cytokines make increased vascular permeability, leaky blood vessels. Chemokines, a subset of those cytokines, are going to mobilize those white blood cells. So at this point, we have the door open, basically, with the leaky blood vessels. We have the leukocytes on their way to the site, but they are actually going to have to get out of the blood. They're using the blood to travel there and get into the tissue for the site of infection. Remember, most infections, some of them will occur in our blood, but generally our infections are going to occur in our skin, in our stomach, and our lungs. And so we need to get those white blood cells out of the blood and into the tissue. And this process is known as extravasation. So extra, think like extraterrestrial, getting out. Vasation, think of vasculature. So how are we getting out of the vasculature and into the tissue? And this is basically a 5-step process. We're going to put notices all over the blood vessels right where the site of inflammation is. We're going to get those white blood cells, those leukocytes that are rushing through the blood, to slow down. We're going to get those leukocytes actually prepared to stop. They're going to stop, and then they are going to exit. So we put up signs, leukocytes slow down, get ready to stop, leave the blood into the tissue. Of course, we have to go into more detail of these steps. What fun would it be if we just stayed there? So the first thing we're going to do is we are going to put up signs. We have blood flowing through our blood vessels all throughout the body. The white blood cells know that they're needed somewhere, but they don't exactly know where. The chemokines and cytokines are flowing through the blood. So when at the site of infection, cells are going to be releasing lots of cytokines. So here, this is a tumor. We're actually going to pretend this is an infection. It's hard to find good diagrams. The cells around the infection are going to be releasing cytokines, shouting for help. And part of that is going to make the blood vessels leakier. But it's also going to induce the expression of ICAM and selectins. Selectins are essentially our slow down signs. And ICAMs are actually our stop signs. So basically, the release of cytokines is going to make the blood vessels right around there near the release leaky, and it's going to have those expressing new proteins. Slow downs, the E-selectins, and ICAMs, the stop signs. And we can see this as soon as we add cytokines, and here IL-1 is an example of a cytokine, we see an increase in these selectins and these ICAMs, these slow and these stop signs. Just a little practice looking at graphs, you can see this is the number of cells. So here there are very few cells that have no selectins. And if we have this group without any cytokines, most of the cells, kind of the majority of the cells, have a little bit of selectin. As soon as we have added the cytokines, though, we're going to have a lot more of those selectins. So a real increase in the number of selectins that are there, triggered by the cytokines. Now, why do we need these selectins? Well, the thing is that blood actually moves pretty darn fast. And so in order, when you're on the highway, you don't just like jump off the exit or stop immediately. Usually there are lots of warnings to slow down before you absolutely have to stop. So you slow down, have some time to get ready, and then can actually stop so everything doesn't just crash into each other. Same thing is occurring with our blood cells. And I'm going to show you a video here. So here we have a video of blood. And what you're going to see when I start this is that things are moving very quickly. All right, gotta stop the annotate. Okay, and annotating. Start. You're going to see how fast everything is moving. But what you're also going to notice is that we have some cells here that are actually slowing down. If you watch these white dots, they are not moving quite as quickly. And this is something that maybe is counterintuitive. I guess I had never thought about how fast our blood was moving and how much it would take to stop. But what is slowing these cells down, what is slowing these leukocytes down, is that interaction of selectins. Now, what are these selectins interacting with? The selectins are the slow sign on the blood vessel. And these are actually going to be interacting with Sial-Lewis X. What is Sial-Lewis X? Sial-Lewis X is actually a carbohydrate that is on leukocytes. So we have an interaction here of selectins on the blood vessel and a carbohydrate known as Sial-Lewis X on the white blood cells. This interaction slows them down. Now, as they're slowing down, the white blood vessels are kind of like, okay, what's going on here? Well, what they're going to get next is a signal to prepare to stop. And that signal to prepare to stop is actually the exact same chemokine or a similar chemokine that got them moving in the first place. As the cytokines and chemokines were leaking out from the tissue, some of them actually stick to the vessel. And that actually gets the leukocyte prepared to stop by activating the integrin. The integrin is just a protein on the white blood cell. When it gets a stronger signal from the chemokines, it will prepare to stop. It will become high affinity, meaning it's going to start, it's looking for that stop sign. And that stop sign then is actually the ICAMs. So we have the selectins causing rolling, basically slowing things down. ICAMs, selectins interacting with carbohydrates on the white blood cell, Sial-Lewis X. We have chemokines that have activated the integrins. And these integrins, also known as LFAs, are going to interact with ICAM on the blood vessel. So we have blood here. We have the leukocyte or the white blood cell, the immune cell here. So we put up the stop signs. We put up the selectins, we put up the ICAMs. As the cell came rolling along, the selectins interacted with the carbohydrates. It got a taste of some chemokines that told it to get ready to stop. So the integrins got in ready, basically in position. So when the integrins and the ICAMs came together, it actually stops the cell from moving. Once the cell stops moving, it needs to get out. Now, this is known as transmigration, just transitory, trans going across. And there are different ideas about how this happens, whether it actually sneaks in between cells, which was what everybody assumed it did for a while, or whether it actually goes through the cells of the blood vessel. We don't fully know. There is evidence of both. You don't need to know all the details, just know that transmigration occurs. That is the last step of extravasation. of a cell being able to leave the blood vessel and get to where it needs to be. So why do we spend so much time on all of this? Well, it turns out that blocking that ICAM LFA interaction, integrin interaction, or blocking that selectin carbohydrate interaction can be great ways to slow down an immune response. Because immune responses are going to build. The cells are going to call for help. White blood cells are going to come in. They're going to be much louder, much more able to do things. They're going to call for backup. A ton of things are going to be triggered from there. And sometimes we don't want inflammation. Sometimes it's going too far. And so we have a number of drugs that will block either, and sometimes even, and I'm sorry, I forgot to mention, even the chemokines themselves, just making that signal not occurring. So blocking all of these steps of chemoattraction can be very powerful when we don't want an immune response. Because if the white blood cells don't get to the tissue, they can't really damage and start attacking the tissue. Now, we want them there if there's bacteria, viruses, fungi, parasites there. We don't want them there if they're accidentally and incorrectly activated. And sometimes they stay activated too long. So turning off the immune response can be very important. And chemoattraction is a great potential place to stop the immunity. And I don't remember if I was going to stay with this slide. Sorry. All right. That's it for convergence. We've now recognized it was a problem. We've alerted some cells. We have gotten everybody to place. The last thing we need to do is actually attack that freaking pathogen in our next video.